if you follow medical science, you'll know cell and gene therapies, CGTs are not just another step forward. They feel like a whole different path.
Absolutely. We're moving away from just managing symptoms.
Right, towards treatments that actually help the body fix itself.
Hmm.
Repairing cells, replacing faulty genetic code. It's really transformative stuff.
Especially for diseases that, we previously thought were pretty much untreatable. Rare conditions, complex disorders.
So our mission today in this deep dive is to get into the weeds of the new safety and regulatory thinking these therapies need.
Hmm.
Because when you're engineering life itself, the systems we use to keep patients safe, pharmacovigilance, PV, well, they need a serious upgrade, right? They have to handle a totally different set of risks.
That's right.
Welcome to DDReg Knowledge Capsule. The materials you shared with us really drive this. Home PV for CGTs isn't just ticking boxes. It's more like building a whole new infrastructure.
It is. It demands very specific methods and, critically, this unprecedented long-term commitment.
Okay, so long-term commitment. How is PV for these living drugs different from, say, a standard pill?
Well, think about it. A traditional drug, a small molecule, your body processes it, and it's gone relatively quickly.
Okay.
CGTs often persist. They're designed to stay in the body, working for years, maybe even permanently. So PV has to cover that whole life cycle.
The entire life cycle, from manufacturing onwards.
Exactly. From the moment it's made, through treatment, and then potentially for decades after. It's about constantly checking that benefit-risk balance, spotting any adverse events early, and this systematic, really long follow-up.
Decades. That sounds like it needs incredible coordination.
It really does. You need this tight, specialized collaboration that you just don't see as much in standard pharma.
Between who?
Between the clinical teams, the regulatory experts, and this is key, the manufacturing side.
Ah, right, because the product itself is complex and variable.
Precisely. The quality, the characteristics of that specific batch of cells or vectors directly ties into safety, potentially years later. Protecting the patient means protecting the product integrity from start to finish.
Okay, let's unpack that a bit more. Traditional PV worries about things like, I don't know, liver toxicity, maybe allergic reactions. What are the specific safety risks unique to CGTs that made regulators say, hold on, the old rule book won't work here?
Yeah, the risks are just different in nature. We have to start way back before human trial even begins in the preclinical phase.
Okay.
We're looking for potential hazards tied to how these therapies work, things that might not show up for a long time.
Like what specifically?
Well, there are three main areas we focus on, really intensely. First, off-target effects.
Meaning the gene edit goes wrong.
Sort of. Making sure the therapy alters the gene it's supposed to, and only that gene. We use detailed genomic analysis to hunt for any unintentional edits elsewhere in the genome.
Okay, that makes sense.
Yeah.
But second?
Second is immunogenicity. Basically, will the patient's immune system react badly to the therapy?
Like rejecting an organ transplant.
Similar idea. We test if the body might attack the delivery vehicle, the viral vector, or maybe the engineered cells themselves, or trigger a wider immune reaction.
So the body fighting the delivery truck, essentially.
Exactly. And the third one, this is where the really long-term concern comes in, is tumorigenicity.
The risk of cancer.
Yes. Because we're inserting or changing genetic material, there's a theoretical risk called insertional mutagenesis.
Okay, break that down.
In simple terms, the worry is that the new genetic material might accidentally insert itself into a spot in the DNA that disrupts a gene-controlling cell growth, like a tumor suppressor gene.
And that could lead to cancer developing years later.
Potentially, yes.
Yeah.
That's why those long-term animal studies are done, to assess that risk.
Right. This is where it gets really complex then. Once these therapies actually get into human trials, the clinical monitoring must be completely different too. What kind of immediate acute safety issues are PV teams looking for right after treatment?
Yeah, in that immediate clinical phase, we're watching very closely for specific, sometimes severe, reactions that are pretty unique to CGTs.
Such as?
A big one is cytokine release syndrome, or CRS.
I've heard of that. It's like an inflammatory storm.
Exactly. It can be a really severe system-wide inflammation as the immune system goes into overdrive reacting to the therapy, potentially life-threatening. We also watch for neurotoxicity effects on the nervous system.
Okay.
And particularly if the therapy involves cells from a donor, we monitor for graft versus host disease, GVHD, where the donor cells attack the patient's body.
So these require immediate specialized care.
Absolutely. Rapid intervention is critical.
So if those are the immediate risks happening right after treatment, is the bigger challenge actually the stuff that might only appear, years down the track?
That is really the fundamental difference defining PV in this field. That potential for delayed adverse events, like the tumorigenicity risk we just talked about, means the long-term follow-up, the LTFU protocols, are absolutely essential.
And how long are we talking?
For many CGTs, particularly those using vectors that integrate into the DNA, patients need to be monitored for up to 15 years after treatment.
15 years.
15 years. It's often mandatory for tracking things like how long the vector persists in the body and, crucially, watching for any signs of delayed cancers or other issues.
Wow. Okay, so if you're tracking someone for 15 years after a single treatment, you absolutely must know exactly which product, which batch they received. That seems like a huge logistical challenge.
It's the ultimate traceability puzzle. It demands this seamless link, end to end.
Linking what to what?
You have to connect the manufacturing data, the specifics of that particular batch, how the cells were processed, where they came from with the clinical trial data, and then connect all of that to the long-term safety data from that patient.
So if a problem pops up 10 years later,
You have to be able to trace it back to the precise characteristics of the therapy batch they received. It's critical for understanding and managing risk across the board.
tracking something for 15 years is one thing, but keeping that data chain secure and reliable, especially when tech changes so fast, paper records aren't going to cut it, surely. How do you ensure that link stays intact and trustworthy across different companies, different countries?
You're right. Traditional methods just aren't robust enough for this time frame and complexity, and the sources you shared highlight this emergent tech is absolutely vital.
What kind of tech?
Specifically, there's a lot of focus on using things like blockchain-based tracking systems.
Blockchain. Like for cryptocurrencies.
Sort of, but the underlying technology. The idea is to create an immutable, tamper-proof digital ledger, meaning the batch identifiers, the patient links, they're recorded in a way that can't be secretly changed or corrupted later. It creates this incredibly secure, verifiable chain of custody that can, theoretically, withstand those decades-long timeframes.
Okay, so we've got these really advanced safety protocols, cutting-edge tech like blockchain. But what are the limitations? What hurdles are PV teams still grappling with every day that tech hasn't completely solved?
Well, despite the advances, there are still some fundamental challenges, mostly stemming from the complexity and the scale, or rather the lack of scale sometimes.
Okay.
We generally talk about 4 main limiting factors. First is the sheer unpredictability of the immune response.
Still, even with all the testing.
Yeah, immune reactions can be highly individual. We're getting better, but precisely predicting or characterizing how each person's immune system will react long-term is still really tough.
Right. What else?
Second is manufacturing variability. This is a big one for CGTs.
They're often personalized.
Exactly. Many are made from a patient's own cells or very small batches. So unlike a pill that's mass-produced to be identical, each batch of CGT can have slight differences. Variations in the cell source, the processing, it all potentially impacts consistency and safety.
It's like the difference between mass-producing identical cars and building custom race cars for each driver.
That's a good analogy. Then third, there's the challenge of small patient cohorts.
Because these often target rare diseases.
Precisely. So your clinical trials might only have a handful of patients, maybe a few dozen. That makes it statistically really hard to detect rare side effects or safety signals. Is that one event a real signal or just random chance? It's tough to know with small numbers.
I can see that. And the fourth?
The heterogeneity of adverse events. The types of side effects, when they appear, it can be really variable and often delayed. This means you need really long observation times and, crucially, very standardized ways of collecting and analyzing data across different studies in different countries. Harmonization is key.
Okay, so those are some pretty significant hurdles. Immune unpredictability, manufacturing variability, small patient numbers, diverse and delayed side effects. What strategies are PV teams using to try and overcome these, especially the issues around small sample sizes and long delays?
The core strategy is really about extending surveillance beyond the initial trial into the real world and making it much more systematic.
No.
A major tool is implementing comprehensive post-marketing registries. These collect data from much larger groups of patients after the therapy is approved, giving us better real-world evidence.
Right. More numbers, more power to detect signals.
Exactly. We're also developing very detailed, very specific risk management plans, or RMPs. These aren't generic, they're tailored precisely to the known and potential risks of each specific therapy.
And the follow-up.
Establishing really systematic, robust follow-up mechanisms to make sure we actually capture those delayed outcomes effectively over the long term. It's about building the infrastructure for long-haul data collection.
And technology must play a huge role here, too, moving beyond just tracking to actually anticipating problems.
Oh, absolutely. Digital transformation is critical for making PV more precise and efficient. We're seeing AI and machine learning being used much more.
How does AI help?
It can sift through enormous amounts of data from different sources, clinical data, genomic data, registry data, even insurance claims data, to spot potential safety signals much earlier than humans might.
And what about getting data directly from patients?
That's another big area. Things like wearables and digital health tools allow for more continuous, real-time monitoring of patients' safety parameters while they're living their normal lives.
So moving safety monitoring from an occasional checkup to something more continuous?
Kind of, yeah. And patient-reported outcome platforms are also important, letting patients directly report symptoms or experiences, giving us a constant flow of information from the people actually receiving the treatments.
Okay, let's circle back to those timelines because they really are striking. We've talked about the biological reasons and the logistical need for this long-term tracking, like 15 years. It makes sense that regulators around the world are locking in these multi-decade commitments. How do the big players, like the US and Europe, compare on these requirements?
They both mandate long follow-up, but the details differ, and yeah, the timelines are really significant. In the U.S., the FDA, specifically the Center for Biologics Evaluation and Research, or CBER, oversees CGTs.
Right.
They generally require that long-term follow-up, especially for integrating vectors, typically 15 years. And they often require specific risk evaluation and mitigation strategies, known as REMS programs, for therapies deemed higher risk.
Okay, 15 years in the US and Europe. I
Yeah.
remember you saying it was even longer there.
That's right. The European Union, through the European Medicines Agency, or EMA, regulates these as advanced therapy medicinal products, or ATMPs. They also require mandatory risk management plans.
And the timeline.
The EU standard is considerably longer. They require 30-year traceability and long-term follow-up under their specific pharmacovigilance guidelines, GVP Module XV.
30 years. 30.
30 years. Mandatory traceability of the product and monitoring of the patient.
That is staggering. That's longer than many researchers' entire careers. It's a generational commitment almost.
It truly is an institutional commitment, well beyond any single company or research team. And you see other global regulators like Health Canada and bodies like the WHO pushing for more harmonization in PD practices globally. Everyone recognizes this level of long-term oversight needs standardized, robust approaches.
So pulling it all together, CGTs are this incredible leap in medicine, but they absolutely demand an equally advanced PV infrastructure to go with them.
Exactly. The sheer complexity means you need continuous data-driven safety oversight across that whole life cycle, from the lab bench right through potentially 3 decades after treatment.
The safety system itself has to be as innovative as the therapy.
We have to treat the safety infrastructure itself as a kind of high-tech, multi-generational project. It's not just a regulatory hurdle. It's fundamental to making these therapies viable long-term.
Which really brings us to a final, quite challenging thought for you, our listener. Given that mandatory 30-year traceability in places like the EU, and this increasing reliance on tech like blockchain to ensure data can't be tampered with, how could we possibly guarantee data integrity and, just as importantly, patient privacy over 3 full decades? Especially when the digital systems we rely on, the software, the security, the data standards change so rapidly, maybe becoming obsolete every 5 or 10 years. That feels like a challenge that goes way beyond the medical science. It's about long-term global logistics, data governance, maybe even geopolitics. Definitely something for you to mull over.